So it's a big pleasure to introduce Dr. Thais Enoki. Thais holds a bachelor degree in biological physics from Unesp and a master and PhD in physics from University of São Paulo. She's currently working in the Department of Molecular Biology and Genetics at Cornell University in the US, and here in Brazil in the Institute of Physics at University of São Paulo. In her research, Thais is interested in investigating the fundamental lipid, protein, and peptide-lipid interactions using model bilayers. Bilayer? Do I say bilayer? I don't know. Bilayer, okay. To understand things like the mechanical and structural properties of lipid bilayers and how protein and peptide-lipid interactions induce bending and membrane curvature. Sorry. It's the powder. Thank you so much. And
thank you very much. Thank you for the organizers to invite me here. And today I will talk about unraveling the complexity of the plasma membrane from studies of symmetric and asymmetric model membranes. So the plasma membrane. The plasma membrane is a very complex system. It's a lipid bilayer, and it is formed by hundreds of lipids. It also has proteins, glycoproteins, and and besides that, the bilayer is asymmetric in terms of lipid composition, meaning that the outer leaflet has a lipid composition that is completely different from the inner leaflet. So the outer leaflet is enriched with sphingomyelin, phosphatidylcholine, and cholesterol. And in model membranes, this mixture may form a lateral organization where two phases coexist, LD and LO phase. So according to the lipid-Hoft hypothesis, these domains are important for the cell function. On the other hand, the inner leaflet is enriched with PE, PS, and cholesterol, and these, in simplified models, this mixture forms one single phase, LD phase. So here we will characterize symmetric and asymmetric domains, and we are interested in studying the communication across the membrane and the interleaflet coupling between these two leaflets of different compositions and phase behavior. Yes?
I only want to know what is LD and LO. I'm not familiar with the term.
So I will explain a few slides about these two phases, LD and LO phase. This coexistence of phase is important because it suggested that cells, they have these lipid haftes, and that are these domains. They are specialized domains that are important for the cell function, and they are enriched in cholesterol. And for example, they could explain how proteins are distributed in the membrane. Or for instance, it's suggested that some viruses such as influenza or HIV bud off from these lipid haftes. Okay, so model membranes. We will use these model membranes to study the behavior of the plasma membrane. It's a model. In this model, we do not have proteins or glycoproteins. It's just the lipids, right? And these lipid bilayers are prepared in vitro with a very accurate lipid composition. And these lipid bilayers self-assemble into these vesicles, that is this spherical shell of determined size. Actually, we have different procedures that we can create these vesicles with different size. For instance, we can create vesicles of size of nanometers or of size of micrometers. And for these vesicles, we call giant unilamellar vesicles. And they have size that are similar to the size of the cells. Here it's important to note that these lipids, they have these properties. So they have a hydrophilic head group. So this hydrophilic group does not like to interact with water. And the hydrophobic acyl chains. The hydrophobic, oh, sorry, the hydrophilic head group likes to interact with water. The hydrophobic acyl chains does not like to interact with water. So the head group, that's hydrophilic, shields the acyl chains from the water exposure. So then they can form this bilayer or this spherical shell. Was that clear? Okay. Okay, it's also important to know that lipids, they have a main transition. And this transition is from gel to fluid. So the gel phase is very organized, and they may have a lattice organization. In the fluid phase, the acyl chains are more disordered, and they lost this lattice organization. And here is an example of two lipids. These lipids has 14 carbon on the acyl chains, and they have saturated bonds, so single bonds. And here is the main transition of the MPC. It's about 23 Celsius degrees. For this another lipid here that is called the PPC, it has 16 carbons on the acyl chains, and the main transition is different. It is about 41 Celsius degree. So these lipids, they have different properties, and they have, for instance, different affinity for cholesterol. And it's very interesting that the cells, they have hundreds of lipids, and several lipids that we call high melting lipids and low melting lipids, and cholesterol. And these lipids, they may organize, forming these coexistence of phase or domains. Okay. So here is the lipids that I will use today in my talk. The SPC is a high melting lipid with 18 carbons on the acyl chains, and the main transition is 55 degrees. And these two lipids are low melting lipids, and they have unsaturation in the acyl chains, so that gives that lipid more disorder in the acyl chains. This one, the main transition is -17, and POPC -2, and cholesterol that is also present in the membrane. Okay, so then a mixture of lipids may form different phase. Here is the gel phase that we just talked about. The gel phase is like a crystal phase, and the lipids are very organized, and they have a slow lateral diffusion. The fluid phase, they diffuse faster, right? And here we have the LD and the LO phase. The LO phase is very interesting because the acyl chain is organized as a crystal, but the diffusion of the lateral diffusion of these lipids is like in the fluid phase. And with this mixture here, the SPC, the OPC cholesterol, we may have a rich phase diagram. That in this phase diagram, we have this two-phase region where LD and LO phase coexist. And for example, here, fluid and gel phase. And between then, the three-phase region. So the LO phase. The LO phase is very interesting because, as I said, the acyl chains are as ordered as in a crystal. However, lipids and cholesterol diffuses in the bilayer as in a fluid phase. And if we look at the chemical structure of cholesterol, that is this one, we see that cholesterol has a poor head group. The head group here is just a hydroxyl, sorry. So then, if we look at the cholesterol cross-section, or if we look at the top view, sorry, go ahead.
Hear me? So the phase separation processes that you are mentioning, all of them, they occur on the surface, right? So inside the membrane, the layer, not inside the vesicle.
The vesicle is a spherical shell. And
Okay.
that is a bilayer of lipids. And then there is two different phases that it's formed by, each domain is formed by different compositions.
Mm-hmm.
So one is enriched with cholesterol and high melting lipid, that lipids with this lipid here, with saturated chains. And the another domain, here is an example that what we see in these GUVs. See, the domains are these pink domains, they are fluid domains. And the blue continuous phase here, it's another phase, that's the LO phase.
So the phase separation is on the surface. There is nothing separating inside the...
Inside we have water, right? In
Okay.
these models we have water. like in the cell, we have the cytoplasm, right? So it's in the bilayer. This phase separation is in the bilayer.
Perfect. Thank you.
Just the thing that I don't know if I understood correctly is that in this LO phase, we are like cholesterol enriched, and
Yes.
then it's more fluid. But what
So
is
the
what's
low
meaning?
phase is actually more ordered and
Yeah,
is enriched with cholesterol.
but then it's
The-
diffused like a liquid phase.
Yeah, the diffusion of the lipid and cholesterol, it's a fluid. They diffuse very fast.
But then they diffuse as a group, right? Like the lipid
Both.
raft.
The lipids, they have a self-diffusion, and the domains also can diffuse. And that's it's because that comes the name lipid rafts, because it's
Yeah.
like a raft. It's like a raft in the sea, right? And the proteins can see that raft and may bind into that raft.
Okay.
More questions? Okay. So here, I was trying to explain how the low phase form, because cholesterol has a poor head group. And so cholesterol cannot shield his hydrophobic core from the water exposure very well. But if it is high melting lipid that has saturated chains, organized chains, there is more room to fit cholesterol and and this head group can protect cholesterol from water exposure. So that was called umbrella model, and that's how the low phase is formed. Okay? Okay. So then we have this phase diagram, and as I said, this phase diagram is very rich in this two-phase region or three-phase region. So how to read this phase diagram? For example, here, this composition here of this star, that is 0.4 of DSPC, 0.4 of DOPC, and 0.2 of cholesterol. So this mixture form LD and a low phase, and you can see that the sum of these fractions is equal 1. That's for the entire phase diagram. Okay. So then we have this phase diagram here that I just told you, the SPC, the OPC, cholesterol. And if we prepare samples here in this two-phase region, we see these macro domains, LD and LO domains. If I replace this lipid here, the OPC, by POPC, and I prepare one sample here in this two-phase region, and I look at the microscope, I will see my GUV uniform. I do not see domains. However, we know that there is domains there, but the size of the domains are below the optical resolution. So these domain size are below 200 nanometers. And how do we study these domains? Okay, so in this work, one of our collaborators studied this system using SANS, small angle neutron scattering. And he found the domain size for this mixture here around 7 nanometers radius. But different groups, and our groups as well, found these nanodomains using different techniques, ESR, FRET, nanosins, and SANS. So here, actually, when I arrived at Cornell, we developed a new method to measure the domain size below the optical resolution using FRET. That is important because it's not every lab that has access to a neutron scattering facility. Actually, in Brazil, we do not have a neutron scattering facility here. So, but using FRET, these experiments are performed in a fluorimeter, and many labs have a fluorimeter. So I will explain this FRET experiment. First, it's important to know the concept of FRET. FRET, we have two probes, two fluorescent probes, and if they are close together, the FRET efficiency is very high. However, if they are far apart, the FRET efficiency decay. So we can monitor the FRET signal on this equipment fluorimeter. And here it's also important to know that the force and distance is when the FRET signal, the FRET efficiency is 50%. Okay. So then in these experiments, we prepare several samples along this line here that we call FRET trajectory or trajectory. And it's important to note that this trajectory is on a timeline. So in this two-phase region here, we have a timeline. Okay, and then in these samples, I will have two probes, one that works as a donor in the FRET pair, and another one as a acceptor. If we have one single phase, these probes, they will be randomly distributed in the bilayer. So we have a certain signal of FRET. When the two phases are formed, when domains are formed, we can have two probes that phase separated. It means they like to go into different phases. So the separation distance of the probes increase, so then the FRET signal decrease. And we can also have the second case where these two probes go into the same domain, so the FRET signal increase. So here is the result. Here is the experimental result. So here we have one single phase, so the FRET signal is about constant here. And when we enter in this two-phase region, the FRET signal decays, because in this case I am using two probes that they prefer different domains. And we can fit the data with this equation here. I like these experiments very much because they are very precise and they can determine the two-phase regions. They can determine this phase diagram. That was how this group that I come from in Cornell, determines several phase diagrams with lipids. Okay, and using this equation, we can obtain the partition coefficient of each probe. And the partition coefficient, it's like how one probe prefer more one phase instead another. And to use this equation, we have to input this data here, that is the signal that we measure in the endpoint of the tie lines. Here is the FRET signal from this endpoint, and here is the FRET signal from that endpoint. And in the second example, these two probes, they prefer the same phase. So here is the profile of when they prefer the same phase, and we can also fit the data. So then we use these FRET experiments to determine the domain size that are below the optical resolution. But we have to discuss this concept before. When we have macro domains, the separation distance of this probe is much greater than R0. So, So in this case, that two probes, they prefer different phase, the FRET signals decays. However, if we have domains that are nanodomains and the same order of R0, like in this second case here, even when my two probes are phase separated, they still in the range of R0. So I have to account for the domain size to interpret this data. So we use a different approach here. We use a Monte Carlo simulation. And in this Monte Carlo simulation, we simulate a sphere that is our vesicle. And on the top of the sphere, we distribute domains of a certain size. And we can do several simulations changing the domain size. And then we distribute the dyes, our probes, according to the partition coefficient. We measure the partition coefficient experimentally. So that is inputting our code. And then we calculate the FRET signal from the separation distance between these probes from the Monte Carlo simulation. Okay, so then we test this, our simulations, comparing to the analytical equation. And here we saw a very good agreement. So that validates our code. And using this code, we simulate these FRET signals from this mixture that form nanodomains. And we obtain a radius that is 10 or 7.5 nanometers that is very similar to the size that was found using SUNS data. For a different mixture, we have a domain size of about 5 nanometers. So that also validates our method to measure domain size below the optical resolution. Okay. In this example here, I also use this Monte Carlo simulation to simulate these fret curves, 4 different fret curves. And this fret trajectory is shown here in this tetrahedron, the two-phase region. And, okay, so the results tells us 2 different regimes. One regime that that is independent of the vesicle size. If I change the vesicle size, I always get a nanodomain. And the second regime that we call macroscopic phase separation, or macroscopic regime, if I increase the domain, the vesicle size, the domain size also increases. That's because these fret curves, they are related to the area fraction of the domain. So these 2 vesicles, they have different size, but each domain have the same area fractions, that is about 50% of the GUV. And because of that, the domain size is very different. So we can easily see that if we have GUV with size of micrometers, in this regime, we still have nanodomains. And in this macroscopic regime, the vesicle size, that is microns, gives us a domain size of microns as well. So this domain size transition, I will explain in a few slides, but it's abruptly changed in the domain size. Okay. So then I explain these 2 phase diagrams here, right? This one forms macrodomain and this one forms nanodomains. So now we prepare a sample along this trajectory here. And in this tetrahedron, in this near phase, I have this phase diagram. And in this far phase here, I have this another phase diagram. And so we prepare samples with 4-component mixture along this trajectory here. And this rho is the fraction of this DOPC, POPC. So basically means I will gradually replace this lipid POPC by DOPC. And then that is what I observe along that trajectory. So we have some compositions that form nanodomains. Then we observe modulated phase and then macrodomains. And here is some examples of modulated phase. They are these nice patterns that we observe in the GUV. And we did these experiments for 12 different lipid mixtures. And we always see this kind of behavior, nanodomains, modulated phase, and then macrodomains. However, the compositional range where the modulated phase changes for each lipid composition here. For example, in this first case, we can see that this mixture favors to form macrodomains and do not form nanodomains. And there's another mixture here. We have a region that forms nanodomains, so that it stabilizes more nanodomains than macrodomains. Okay. And we also add one peptide on this lipid bilayer, and we observe that this peptide shifts this row window. We call this row window, this distribution here, is where we observe this modulated phase. And these peptides shift this row window to lower values of rho. So which means it stabilizes more macrodomains now than nanodomains. So this mixture here is actually this one plus the peptide. Okay, so today I will not talk about protein-lipid interaction, but this slide is here just to show when we add proteins or peptides that may change the phase behavior of our bilayer. In this case here, that's the same peptide that I show before here in this case, we observe that these peptides make the LD phase much more stiff, much more difficult to bend than the LO phase. And in this second example here, we add a protein that they have this glycocalyx, and because the interaction of this glycocalyx, they make the bilayer to bend. And from this, the Geophy studies, I don't know if I can play the video again. We can see that for several tubes that come out of the Geophy. And it's very interesting that this protein that's called podocalcin is overexpressed in in some kinds of cancer. They always observe that in cells. Okay, so then I will return to the measurements in lipid bilayers. So here without proteins. Okay, here we measure the line tension using flicker spectroscopy. So line tension, you can understand line tension as the energy penalty here when we have two different phases touching each other. And we can measure line tension. In these experiments, we measure the line tension for these two morphology when we have macro domains or modulated phase. And here we apply a simple trick that we cool this sample very quickly, so that helps to form these domains with a smaller size. We need a domain with a size of about 5 micrometers, and then we record the fluctuations of the domains, and then we can calculate the line tension of the domain. And we observe that the line tension of this morphology that forms a modulated phase, it's lower than this case. But this result is a kind of obvious, because if we have a very high line tension, that means that tends to minimize the perimeter of the domain. So that tends to form these macro domains, one single domains and macro domains. If the length that these two phases are touching each other, so if the length increases, so the line tension can decrease. Then we measure the line tension for these 6 different lipid mixtures. And for this one, palmatoid sphingomyelin, DOPC, POPC cholesterol, we measure the highest line tension. And we correlated that with the row window. We observe that the row window is shifted to lower values of rho. So it means that favors the formation of macro domains. So that means a higher line tension. And for this another mixture here, that is brain sphingomyelin, DOPC, POPC cholesterol, it has the lowest line tension. And that is the row window for that mixture. We can see that it stabilizes more nanodomains. So we can use the position of this row window to give you a guess about the line tension. Although that does not give us a number for line tension, we can use that to compare to different systems. As for example, this system without the peptide and with peptide. When I add the peptide, the row window shifts to lower values of rho. So that means it raises the line tension. It favors to form macro domains. Okay, so along this domain size transition, we measure different properties of the membrane, as for example, the bend modulus of the LD and the LO phase. And they do not change much. We observe a change that's about 2-fold, but this changes because I am changing the composition DOPC by DOPC. Another experiment, we measure the partition coefficient of this lipid. This lipid has a spin label here, and the partition coefficient does not change much. And we also measure the order parameter of the LD and the LO phase along the main transition, and we observe that also does not change much. So in this plot, we summarize the data that we measure. For the domain size, in this regime, we have nanodomains, so domain size of nanometers. And in this regime, we have domain size that is, there is a domain size transition that these domains, for example, have size of micrometers. Any other parameter here, as band moduli of LD phase, LO phase, do not change much along this transition. And the other parameter are partition coefficient. So there is no abrupt transition in these phase properties, but the domain size change a lot. So to explain this result, we propose a competition of interaction. And this is the competition of the line tension and a dipole-dipole repulsion. The dipole-dipole repulsion, we do not know exactly what that comes from, but we suggest that could be from this carbonyl group, because this bilayer, they are packed, they are oriented, right? So that could rise these dipoles. In this model, we observe that the total energy can be minimized for a certain domain size that is about tens of nanometers, and that could explain this abrupt domain size transition that does not have a hallmark of a main transition. Our suggestion was inspired in this work here, that they proposed this competition of interactions to explain this modulated phase in several different systems. Right. Okay, so I return to the first slide. So far we studied this one leaflet of our bilayer, but the another leaflet has a composition that's completely different from this one. This one may form these domains, LD and low phase, but the opposite leaflet may form a single phase that is a LD phase. So we are interested to understand the communication across the membrane. To do that, we first need to develop a method to make asymmetric bilayers. And we did develop one method based on the hemifusion. So the hemifusion is when the two outer leaflets are connected. And when they are connected, they can exchange lipids by diffusion. This work was published at Biophysical Journal and invited to be the cover of the journal. Okay, I will briefly explain the procedure. So we prepared giant unilamellar vesicles, GUVs, with one single dye, red, and a supported lipid bilayer with a different dye, a green dye. Then we established contact between the GUV and the supported lipid bilayer. We add calcium to initiate the hemifusion. Then we monitor the hemifusion on the microscope, so we can see that that GUV that was before only red becomes green. And then we add EDTA to chelate the calcium. And we apply a shear force to detach this GUV from the supported lipid bilayer, and we study these free-floating GUVs. Okay, we did some controls to prove that these asymmetric GUVs are really asymmetric. So in the symmetric bilayer, we have a fluorescent probe, a dye, that is red, that labels both leaflets. In my asymmetric vesicle, the lipids from the supported lipid bilayer and the dyes migrated to the outer leaflets of the GUV. That is what we suggested. So we then add a quencher, a fluorescence quencher, to quench the fluorescence of the outer leaflets. And we observed that for 75% of our AGUV, all the green fluorescence was quenched. However, for 25% of AGUVs, we found a small contamination of this green probe on the inner leaflet of the AGUV. We also did some controls that these asymmetric GUV, they preserve the vesicle content. So we can prove that they are a product of hemiffusion. And this slide is a result, but it's here as a control as well, because these results also prove that these bilayers are asymmetric. So the idea top fluor PC, they are different probes. And both probes, they prefer the LD phase. That's the LD phase. And top fluor PC labels only the outer leaflet. The ID labels the inner leaflet. And in this example here, we have the LD phase opposed to a LO phase. This result is very important because it was once suggested that the interleaflet coupling is so strong that this kind of result was never observed before. So we show that these domains are not aligned, and that could be related with a low coupling strength. So later we start to understand that the anti-registration of the domains are related with interleaflet coupling. In this paper is a theoretical paper, and it shows how to assemble these phase diagrams. And they have a term related with interleaflet coupling. And as they change this term here with interleaflet coupling, the phase diagram change. And it's very interesting that this theoretical paper, this three-phase region here, actually predicts this behavior that these domains are not aligned. Okay, so I will build my asymmetric bilayer. So one leaflet phase separating to LD and a low phase, and another leaflet, the opposite leaflet, is a fluid phase. And here is the composition of the inner leaflet, and here is the composition of the outer leaflet. It will be the composition of the supported lipid bilayer. Okay, so here it's important to note that to build that model that we want that one leaflet must form a LD phase, we need a higher percentage of lipid exchange. As I replace the composition of the outer leaflet, the composition of the outer leaflet changes along this arrow here. So for 30% of lipid exchange, we have this composition on the outer leaflet. And for 75%, the outer leaflet is now a fluid phase. And in this case, we're still observing domains. Remember here that this green dye labels only the outer leaflet. But these domains, they are a little bit different from the symmetric case. And we observe the partition coefficient of this probe, and the partition coefficient decrease. Then we investigate the lipid packing order of these domains. And to do that, we use this probe called Sillardon. And Sillardon presents a spectral shift according to its environment. So when Sillardon is in an ordered environment, that shows a blue shifted. And when Sillardon is in a disordered environment, the spectrum is red shifted. And then we use this parameter here, GP, to measure the lipid packing order of these two phases. For the LO phase, that parameter is positive, and for the LD phase is negative. So we do that in the microscope. So we do a spectral image of Sillardon. And here it's a GUV, and here it's an asymmetric GUV. And then we can compare the order of symmetric and asymmetric GUVs. We observe that for asymmetric GUVs, both domains becomes more disordered. And the difference in GP decrease. So that means these domains here in asymmetric GUVs, they are more alike in terms of order. Okay, It's also interesting that we observe these induced order domains in different lipid mixtures, and this mixture here forms a gel phase. So the inner leaflet is phase separated in gel and fluid, and the outer leaflet is a fluid phase, but also became phase separated because the inner leaflet induced the phase separation. And we observe that for brain single myelin DOPC cholesterol and for DSPC DOPC cholesterol. But I also should mention that before us, different groups have observed this phenomena that the one phase separated leaflet can induce domains on the opposite leaflet. The first group to observe that is a group from Lucas 10, using supported lipid bilayer. Okay, so then if you want phase separated leaflet, if this order domain can induce order on the opposite leaflet, that is a fluid leaflet, we can ask what's happened when we have cholesterol. When this lipid mixture has a high melting lipid, we know that cholesterol likes to interact with this lipid, and that forms the LO phase. In the absence of a high melting lipid, cholesterol may not induce such higher order. However, it does not need to, because the order is induced by this domain here, this order domain. So it's reasonable to suggest that cholesterol may prefer this induced order domain because it packs better with these somehow organized acyl chains. And in other words, there is more room to fit cholesterol, and then to this red group helps to shield cholesterol from water exposure. So then we look at our data to see if we can find evidence that cholesterol may be enriched in these induced order domains. So let's first look at the order domains of an asymmetric Geophy. Here, Silaudan, that's the probe that we are using, Silaudan labels both leaflets. So here is the spectrum of the LO phase, and here is the spectrum of the LD phase composed of DOPC and cholesterol. And we can build this spectrum for an asymmetric bilayer. So it's very important to note that so far we have analyzed GP. And GP is a kind of normalized parameter because it's the intensities measured in two different wavelengths, but for the same spectrum. And here we are going to bring a new information that is these intensities here, and this intensity will give the right weight to build the composite spectra here in blue. So then we compare this composite spectra with this spectrum that we measure experimentally for this asymmetric GUV, and we observe that they are different. The experimental spectrum is blue shifted. So blue shifted means more ordered. So that could suggest that some cholesterol from this domain here moved from these order domains. But that can also suggest that the LO phase induce order on the opposite leaflet without losing so much order itself. So then let's look at the LD domain here. Again, Silaudan labels both leaflets. Here is the spectrum of the LD phase. Here is the spectrum of DOPC cholesterol. The spectrum of the LD phase is a little bit more ordered because it has a small fraction of DSPC. Then here's the spectrum that we built for the asymmetric bilayer, and then we compare with the spectrum that we measure experimentally. And we observe that the experimental spectrum is red-shifted, and red-shift means more disorder. And also, they have different shapes, but we will compare the GP parameter here. So the GP parameter for the composite spectrum, for the spectrum that we measure experimentally, and it's very interesting that this is GP value. It's very similar to GP values of a pure POPC bilayer. So, meaning that this LD domain becomes depleted in cholesterol. So then we suggested that these induced order domains become enriched in cholesterol. Okay, why that is important? Because one leaflet that phase separates may induce a domain on a fluid leaflet, may induce order on this fluid leaflet, but also may induce a redistribution of cholesterol. And if we have a more realistic model for this leaflet here, the inner leaflet, in this case, we use only the OPC cholesterol. But if we have another lipids as PE and PS, that could create a reorganization of these lipids, forming a domain with distinct lipid composition. Here is another example. It's been suggested that viruses such as HIV, they bud off from haftes. However, how could these GAG proteins interact with the leaflet that is supposed to be fluid? But we will observe here that with this mechanism, there is domains enriched in cholesterol, and that could explain better these results, that these GAG proteins could assemble on this inner leaflet than to bud from the lipid haftes. So, conclusions. The exoplasmic leaflet of the plasma membrane can be modeled by a ternary mixture or a quaternary lipid mixture, namely high melting lipid, low melting lipid, and cholesterol. Depending on the lipid composition, we observe macro or nanodomains. We propose a new and accessible method to measure domain size below the optical resolution using FRET. We found nanodomains with size of about 20 nanometers diameter. That's consistent with the domain size measured by SENS. We investigate a domain size transition that does not have hallmarks of a main transition. We propose that competing interactions could explain the domain size transition. We propose a new method to prepare asymmetric GUVs using hemifusion. We can select GUVs that approach 100% of lipid exchange. Exchange lipids are found on the GUV outer leaflet. Hemifused GUVs preserve vesicle content, and one phase-separated leaflet can induce domains on the opposite fluid leaflet. Asymmetric domains are more alike in terms of lipid packing order, and the induced order domain is enriched in cholesterol. So I would like to thank you all for your attention. I'd like to thank my advisors, especially Professor Feigenson and Professor Hebeli. I'd like to thank all that I interact along my trajectory, and I would like to thank the organizers for this invitation. I also would like to advertise that I am organizing this special issue, and if you are interested in lipid bilayers, yeah. Thank you.
Questions?
First, thanks for the seminar. It was really great. Your biological model is a micelle, right?
No, it's not a micelle. A micelle does not have an internal volume.
Oh, yeah.
So it's a vesicle.
Okay.
So it's a bilayer, and there is water on both sides of the head.
But then
A micelle, it do not have the content, the water content.
okay, but then all the process of making this domains is being totally passive here, right? Just by the interactions among the lipids.
Yes.
But in the real cell we have like lipase, flopase, and all this part that is really active. So how can we correlate this, all the experiments that you guys performed, showing how this thing is created by a passive way, when in the real cell we do this in an active way and really.
Right. In cells, we have these flow pace, flip paces, crumb base to maintain the lipid bilayer asymmetric. And here, we do not have these proteins. So we assemble two different leaflets with different compositions, but that it's not in equilibrium. We call a metastable equilibrium. That is in equilibrium for a few days. After 2 weeks, we lose the asymmetry because the lipid flip-flop. So lipid flip-flop happens, but it takes, when we do not have these proteins to assist the lipid flip-flop, that can take some days, a few days. More questions?
Just another really quick question. When you do the hem fusion, you need calcium, right?
Yes.
But this is heat dependent as well, or not?
Sorry, say again.
Heat dependent, because generally.
Yes.
And how will it affect the saturation of the lipids, or doesn't affect?
So we do not have a careful study using the temperature, but that is our next step. But that sure affects the how to create these asymmetric vesicles, and also the lipid diffusion, right? Because they will diffuse a little bit faster. Exactly. If you increase the temperature, right?
Okay, thanks.
Great questions, thank you.
Yeah, I guess I have a simpler question. When you present that that idea of competing interactions for explaining the domain sizes, I didn't understand. Is that a specific model you're using, or are you inferring these interactions somehow?
No, we propose a model. Let's see. That is the line tension and basically the dipole-dipole repulsion here. And maybe I can show the equation. Right, so you cannot see that. Right, okay. Select it this way. It may take some. Okay. So for the dipole repulsion, it's here, right? Oh gosh, here. It's actually a very simple model, and I think everyone that do electromagnetism can do this calculation. It's actually very simple. I have that in my notes as well. Do you have?
It's a calculation based on electrical potential that you're.
Yes.
What is,
let's see if I can open something else here.
I don't think we need to even go into details. I mean, is it really simple? Just.
Oh, the problem is that I don't know how to. Okay, so it's like we have this, that is a low domain, right? And we propose a difference in dipoles from this LO and LD phase. And then, yeah, unfortunately, I cannot show that from here. Yeah, probably most of you know this equation here, right? Yeah, I'm not sure how much detail you want, so it's
No, yeah, I guess all I wanted to know is that this is just like a particular calculation that you can do considering the electrostatics of this system, and you're considering that these, you're not modeling the molecule itself, it's just one dipole actually. It's like you're treating the entire structure as like a dipole that is
Yeah,
a
actually
dipole density.
we are giving a dipole to a domain. To a domain. We are not treating each molecule separately, no.
Ah, okay. So and then so the domain formation here, does it is all local, right? So there is no dependence on the global topology, like the length scale set by the size of that sphere, which is the vesicle, is not relevant for these for these domains, for the size of the domains.
Well, that depends on the regime that we are. So now, could you help me to set this one again?
I think back and forth.
I messed it up.
So you want to go back full screen or?
Yeah, I think we have to set Sorry, eu acho que a gente tem que arrumar aqui como que esse aqui nesse. Assim, mais ou menos assim. É isso que vocês estão vendo. Ok. Só isso, eu acho. I'm not sure if I understood your question, but as I told you, there is two regimes, right? And one that the vesicle size and the domain size are, there is a dependence in domain size and vesicle size. But that is because in this lipid mixture, we have a high line tension. So the domains tend to coalesce in one single domain. And that is related with, with the area fraction. So actually this data from FRET is related with the area fraction of the domain. But I'm not sure if I understood your question correctly.
Yeah, so in this plot, I guess my question is, the only effect that the vesicle being like a finite size is imposing is just that there is kind of an upper bound to the size of these domains. Or is there like something deeper than that?
I think that depends on the interaction that we have. It's because in this case, we have a higher line tension. Maybe if we have a different lipid mixture, we'll have a different result. So, yeah.
Okay. Thank you.
Yeah, I think basically I had the same question, more or less. So the question is why coarsening stops, right? Because you should expect, in principle, if you have short-range attraction and short-range repulsion, that the molecules would clump together up to one single domain, as you just showed in one of the cases. But then, actually, in most cases, you measured microphase separation. So basically, you have that the domains stop growing at some point. So Ostwald ripening stops at some point.
So
and this is because of the long-range dipole-dipole attraction? Repulsion.
There are two, the dipoles also play a role on that, but we also have the line tension. So if the line tension decreases, that may break these domains in smaller domains. So we may also have a different, if I see examples here, no. Do not bring a nice example. All right. So in that case of that GUVs with a lot of large domains, right? That probably means a lower line tension. But we also have some dipole repulsion that breaks these domains in smaller domains.
Yeah, because in principle, if you did not have this long-range repulsion, then they would coalesce into a single domain, right?
If I don't have a long, sorry, say again?
Like, if the domains not repulse themselves, then you would have one single domain in the end.
Yeah, but it's not only the dipole-dipole repulsion. We have to include the line tension. The line tension is always there. and that may change. Is actually the balance between the dipole repulsion and this line tension.
Yeah. Great, thank you.
Can you go to the slide of the induced order phase?
Yes. So which one is this?
You can pass. You can pass the one that have the cholesterol. Yes, this one. Back. Come back. Yeah.
I'm not sure which one. Because I think
Okay.
in
Yes,
the
this
very
one, the
end
big
we
one. Yes,
have
this.
it. Yeah.
Thank you. So if I understand right, the order phase induce order domain in the LG phase, right?
Yes.
Because of the quantity of cholesterol. They push the
Because-
quantity of they push cholesterol.
So the first this domain here induce order on the opposite leaflet. That is a fluid leaflet. But then cholesterol may have a higher affinity to this domain here because these acyl chains are more organized. So it can pack better cholesterol. And as I told you before, cholesterol has a poor head group. So that is good for cholesterol because it offers more protection to cholesterol and shield cholesterol from water exposure.
Okay, so the cholesterol is not there because of it's not the cholesterol that is causing the order. The cholesterol go there because it's ordered. Right? This?
Both. Cholesterol may also induce more order once it's there, because that is what cholesterol does. But it's very interesting that cholesterol may change a lot a high melting lipid. But for this lipid that we are using here, that's the OPC, is not a higher, so much change. Cholesterol has a very interesting effect because it may change more one lipid than another. And this lipid here, the OPC, is a very fluid lipid. And cholesterol has low effect on the OPC. But it can induce some order.
Okay. Because I was thinking the quantity of cholesterol in this layer is limited, like you have a quantity in the model, for example, because if this part is pushing the cholesterol to there, maybe the other part would have less cholesterol and
Yeah. That we suggest that this other part has less cholesterol.
than the usual. And this
Yeah.
could cause like more disorder than the
More
usual.
disorder, yes.
Okay.
But your point is very interesting because so far, research, they do not agree each leaflet has more cholesterol. And it's very difficult to track cholesterol because cholesterol moves really fast on the membrane. And the flip-flop of cholesterol is on the rate of milliseconds or microseconds. So it's very difficult to track cholesterol movement.
Thank you.
Anyone has more questions? Amanda?
I don't know exactly if it is a question, but in the last slide you showed something about various binding and releasing, and I was thinking like, could we can, now that we can actually create these GUVs that have the size of a natural a cell, can we try, I don't know, to enhance our understanding of how these various binding?
That is one of my projects. Yeah, it's what we are gonna try to do next. Thank you very much.
Anyone? No? All right, so let's thank Thijs again for a great talk.
Thank you.